Hello, my name is Kalunda Anthony, and I will be presenting on the effect of age on locomotive control of ELAV-Gal4 times UAS polyQ Drosophila melanogaster. The crossing of male ELAV-Gal4 flies with female UAS-Q128 flies creates a model for which Huntington's disease can be studied. Huntington's disease is a neurodegenerative disorder in humans characterized by the number of repetitive CAG repeats one contains in their genome. In the case of the flies, ELAV is an RNA binding protein that serves as a neuronal marker. Gal4 is a protein activator that binds to and activates the UAS promoter sequence, allowing for the expression of polyQ. The presence of ELAV binded to Gal4 ensures that the expression of polyQ occurs only in the neuronal network of the chosen fruit flies. Being small organisms with only 4 pairs of chromosomes, Drosophila melanogaster are model organisms that can be utilized to study the progression of Huntington's disease. These studies contribute directly to the research of pharmaceutical companies as they aim to alleviate the symptoms of this disease. The goals of this experiment include creating a viable model to study Huntington's through the successful crosses of 2 types of fruit flies, to conduct a negative geotaxis assay to assess the differences in locomotive control of flies of different ages, and to draw conclusions from that collected data that provide new insights into how experiments like these could be improved for the future. Figure 1 displays the possible genotypes and phenotypes expected from crossing male ELAV-Gal4Y flies with female flies that are homozygous UAS-Q128 at chromosome 3. Only the flies whose genotype contains both ELAV-Gal4 and UAS will express Huntington's disease. Figure 2 displays the male and female flies used in this experiment. During its course, the male and female flies had to be separated. Graduated cylinders, tape, markers, and gauze plugs were the excess materials needed to conduct this experiment. 2 crosses were performed to create 2 separate age groups, one between the ages of 7 and 14 days, and the other between the ages of 14 and 21 days. The efficiency of the cross was to be determined by scoring the flies, aiming for a ratio of 1:1 of unaffected male flies to afflicted female flies. A negative geotaxis assay was to then be performed and the resulting data to be plotted on graphs for better visualization. The unaffected male flies were to be used as the control, and the female flies were to be used as the experimental group. Finally, conclusions were to be made post-data collection to qualify the effective age on Huntington's disease-afflicted fruit flies. The results from the 2 assays were very different. In the 1 to 2 week Drosophila assay, it was discovered that the unaffected male fruit flies were more locomotively challenged. Figure 3 shows the data collected as a scatter plot of the number of fruit flies that passed the threshold point out of 10 flies versus the trial number out of 20 trials. The data shows that female flies were consistently able to pass the threshold, but the males had much difficulty. The reverse is true for the 2 to 3-week-old flies, as the afflicted female flies were consistently unable to pass the threshold, while consistent high numbers of male flies were. Figure 4 displays the similar data as Figure 3, but is for a different age group. In Figure 3, the orange line represents the experimental female flies, and the blue line represents the control males. In Figure 4, the yellow line represents the experimental females, and the gray line represents the unafflicted males. Based off of the data presented in Figure 4, the inability of the female fruit flies to consistently pass the threshold is indicative of the negative effects of Huntington's disease on locomotive control. By comparing the data from the 2 figures, it becomes evident that the female flies that were 1 to 2 weeks old were not as locomotively impaired. This suggests that the point at which Huntington's disease significantly begins to affect locomotive control occurs at a point between 1.5 and 2.5 weeks. For future experiments, it would be beneficial to specifically restrict the ages of the fruit flies so that they aren't broad ranges. This specificity will allow for a more accurate timeline of locomotive degradation in Drosophila due to Huntington's disease. Additionally, scientists will be better capable to pinpoint the time period in which locomotive control decreases significantly. This age correlation can then be somewhat applied to the studies of Huntington's disease in humans, as scientists aim to discover the time frames in which Huntington's disease progresses significantly. Thank you.